So I'm going to try to get you to coffee break as efficiently as possible. I'm going to try and do that by standing still and saying less rather than speaking very, very fast. So we're about to find out how successful I'll be at doing that, but I'm going to do my best. Okay, so here we are. Yep. So can modeling accelerate biologic drug formulation? First of all, can we model drug formulation? And the answer is a resounding yes, absolutely. We'll get to how fast we can do it. But we do. We can and do it all day long, and we love to do it. And by we, I mean the royal we. So our modeling team collectively. And yeah, we can model these rough beasts from atoms all the way to processing. So we started our omics, we have our genomics and our proteomics, and that's now being souped up by machine learning, artificial intelligence. My students, Jack and Shubham, do their best to teach me about these things, and I do my best to keep up. Thank you guys for your patience, I do appreciate it. And we also heard from Kevin Kavanagh yesterday about how proteomics can really, really underlie our efforts in drug discovery, our efforts in drug formulation. So proteomics is our starting point. Then we make our atomistic models. We heard from Sally and from Sarah yesterday. You get the right answer with quantum mechanics. You get the right answer at 0 Kelvin for a few hundred atoms, which is brilliant. We can build our force fields from that. So that's what David Tiana's group do down in Cork. Also Rob and Rhea up in DCU do a version of this where they try to map full DFT onto semi-empirical methods. So it gives you a faster method of scanning. And it also gives you access to larger systems. So you can then build your force field, and then you can start to look at bigger things. This is what Cheyenne and Pierre in my group do brilliantly. And you can fit these large models of large proteins, large biologics, to say things like SACs and DLS, which we heard about yesterday from Mikal. So we saw how when you're making clusters of molecules and solutions, you get complexity. You also get a lot of complexity when you look at biologics. So Pierre and Cheyenne spent happy afternoons fitting these creatures to different types of SACs envelopes, which is a very pleasant and rewarding activity. I can personally vouch for that too. So then, depending on how patient you are and how big your computer is, and we do have big computers and we are reasonably patient most of the time, you can actually, with full atomic resolution, start to look at how proteins crash into each other. Or else you can say, to hell with it, do what Sousa does, pull back your microscope and just make bead models. So that way then you can actually get to the mesoscale. You can look at, say, lots and lots of antibodies flocking together in solution in a sea of excipients. And you can get an answer which will still be interesting to industry by the time you actually get the answer. And she does that brilliantly. So by mesoscale models, I mean you map from quantum mechanics to classical mechanics. That's just calculating F equals A, MA billions of times. That's literally what we do. Then you go to bead models. So you don't have full atomic resolution anymore. You treat, say, your amino acids as lumps rather than you lose some of the atomic resolution. But that works remarkably well as long as you carefully parameterize it properly. So it's a continuum, and you build knowledge as you go. And that also gives a shot then to feed into much larger things. So we heard about that yesterday from Norma, what Harry does with the bioreactors, what Norma and her group do with maths and stats. We can map from atoms all the way to processing. And data is ubiquitous here. It's what links these different hierarchies. It's what actually it flows down and it flows back again. It flows back up again. So that gives our link with industry, and data is absolutely central to everything that we do. Okay, so we can model biologics, but yeah, can we accelerate biologic drug formulation? So can we do it fast enough so that, as I say, the answer is still interesting by the time we get it? So that's where the center effect kicks in. And we're not precious about our models. We don't care how pretty they are. We want to actually stress test them. We want you guys to take our blueprints and try them in the lab. And we push that we want to be predictive modelers. We want to actually make predictions which are experimentally testable and see how they get on in the real world. You know, that's where the fun is. If we're right, we're geniuses. If we're wrong, we've got our next project. It's a win-win situation. We're very happy to send them out there. We say they're pretty, but you know, we don't mind poking them around and seeing how they get on in the lab. That's the fun part for us. So in terms of challenges then, we're focused on challenges 1 and 2. So looking at biologics, looking at peptides, antibodies, trying to accelerate their formulation using modeling. Biologics, as we heard from Sarah Hudson yesterday, the point was very well made that biologics can't go everywhere. So small molecules will always have their role in drugs. That's clear. But where we can use biologics, they have unique advantages. So we have natural bioavailability. They can help with solubility. They can help with, you get new ways of doing targeted release, targeted delivery. And it also, obviously, it's greener and it's more sustainable. It gives you a shot at precision medicine. And yeah, the challenges then, of course, are very different for small molecule versus large molecules. So in terms of stability, in terms of manufacturability, there are new challenges for us there, and we can address those by modeling combined with experiments. So the jump in size is daunting. I mean, monoclonal antibodies are 1,000 times larger than aspirin. So even what we usually think about drugs, we think about something burrowing through the cell membrane, finding the right receptor inside the body, say an elliptosine molecule hops onto a mutant c-KIT kinase as an anti-cancer drug. For monoclonal antibodies, they generally don't go through the cell membrane, and not without a lot of work, not without a lot of extra expense, a lot of poking around. So generally, when we talk about antibodies, we're talking about new mechanisms. So here, for example, Herceptin, which is an anti-cancer drug, anti-breast cancer drug, binds to the HER2 receptor on a cell. It switches off some intracellular signaling pathways, interferes with apoptosis. It also actually wonderfully manages to target that tumor cell for elimination by the immune system. So you get all the wonderful complexity and opportunities of evolved biological nanomachinery. So you're not just talking about lock and key mechanism anymore. You can actually interfere with these cell signaling networks. So that's a challenge for us to understand that, but it's also a fabulous opportunity to actually create new types of therapies. Okay, and of course the big problem with biologics is that they're fragile. It's very hard to actually store them, to deliver them inside the body. They tend to fall apart very easily. So we're developing methods to actually cloak the biologics. So that's just putting them in, using molecules to form shells around them and protect them from harsh environments. So it's very simple. You're basically cloaking and hiding the biologic. And the new physics and new chemistry that we're learning is helping us down here too in terms of peptides. So we can learn more about how to, say, create permeation enhancers to try and get peptides through cell membranes. Out here, it also gives us a shot at actually looking at, out in the realm of bioprocessing. So these massive creatures, can we actually trace all the way from the molecular level through to bioprocessing? And this type of knowledge that we're building up is giving us access now also to those areas using multi-scale molecular modeling. Okay. So yeah, just in terms of drilling it down to operational level, if you're looking at, say, monoclonal antibodies, what do you have to look at? So to make a stable formulation, you can look at excipients. We do that. We're working with Sanofi to design basically mixtures of excipients that can protect particular antibodies. For oral administration, if you dare to actually try to make them orally administrable, I would say that's the holy grail. That's not something we're going to solve today or tomorrow, but it is something to aim for. You could try, for example, cloaking at low pH. In terms of efficacy, then you're targeting particular cell types and particular transmembrane cell receptors. So you get a lot of opportunity to work with clever membrane X-ray crystallographers to try to actually resolve what those membrane proteins look like in the first place. In terms of peptides, Sarah mentioned this yesterday in her talk, we look at mechanical strength. So can we actually make more robust, more tolerable formulations? Can we co-crystallize those, say, with cofactors which may help to prevent their aggregation, which may aid their solubility? We also look at, say, for example, different types of peptides. People look a lot at cyclic peptides because they tend to be just naturally very strong, but they're very hard to engineer, and they're also very expensive. So can we make linear peptides, which also actually are robust? And with peptides, then if we can get them through the cell membrane, we can use everything we know in terms of small molecule drugs, in terms of lock and key inhibitors and polyagonism. And polyagonism is either very exciting or very scary, depending on your mindset. What it actually means is that you're targeting two receptors at the same time. And this is Shubham's work. I'll just briefly show you, just give you a very brief flavor of what he's doing. So the idea is here that you're making a peptide drug which will bind to two receptors at the same time. So it targets two of these GPCRs. You've got your glucagon receptor and your glucagon-like receptor. So most diabetes drugs target just the first one, and it tends to trigger obesity. So if you have something that binds two at the same time, you get your treatment for diabetes while also offsetting obesity. So it's a very, very attractive notion. So basically the idea is here that we can provide coagonists that target both of them simultaneously to provide better control of blood glucose and obesity than currently marketed monoagonist therapies. So yeah, we want to basically design more effective combinatorial hormone therapies. And all credit to Shubham for the work that went into this. It spans from informatics all the way to experiments, and there's a hell of a lot of molecular biology in here too. In a nutshell, what he does is he takes his protein structure. A lot of these aren't in the protein database. You have to build them using structural homology and some informatics. He encases that in a large box. He adds his phospholipid bilayer. He's his peptide protein complex, and he puts that in a big, big box of water. And he just calculate F equals ma billions of times. So all in all, his data set is, I think I have it here somewhere, yes, 2.3 microseconds of free dynamics. That sounds like a very short period of time, but that's millions of statistically distinct snapshots of the system. X-ray crystallography gives you one snapshot. So you can watch how the system moves over time. To paraphrase, 5 minutes to wiggling and jiggling of atoms. From that then you can calculate properties. So you can calculate the binding of the different peptides to those receptors. And he has this hit, which is this MDD GLP-1R. We have to think of a snappier title for that. But it's very, very promising because it binds more strongly than codoutide. So it seems to get the best of both worlds in that it binds significantly strongly to both of those receptors. So it's an interesting prediction from the modeling. And we're at that lovely phase where we're actually ahead of experiments. So, we can be brave and put this out there, and we'll see how we get on in the lab. So just to say that it is multi-scale modeling, starting from informatics, then building your molecular dynamics simulations, then mapping that onto machine learning analysis. That's the last part of his PhD, and that's the most challenging part. To actually, once you have the really detailed atomic models, and then try to actually map that then onto a more generic model that can be applied to lots of different proteins. That's a challenge, but that's something we're working on. I'll skip. Just to say he is actually currently working on a triple agonist, which is very fun. Just to make the point that we are aware that there are a lot of experimental challenges here in terms of formulation. And that's where we need the experiments, that's where we need the center effect. We can only do so much with our big computers. So we need to look at, for example, binding assays, we need to look at stability, protection against proteolysis. There's lots of things you have to get right if you want to make one of these drugs. So watch this space. So what we're always trying to protect against when we're designing biologics, or even designing small molecule drugs that go into the body, which obviously any good drug should go into the body, is side reactions. So when you swallow the pill, it breaks down, and then it goes through your body, and it meets an awful lot of stuff on its way to its target site. And we're actually at the stage in modeling where we can model these big, complex systems. These kind of Monet-type paintings were done by Michael Fake. These are just basically showing how many atoms you can cram onto into your box when you have a really, really big computer. Thankfully, it boils down to just looking at interfaces. So you identify, the important interfaces in the system, and then you really drill down and look at those and do full, statistical mechanics treatments of those, measure the binding energies as accurately as you can. So that gives you a shot at finding big things. So in terms of where the field is going, then you're getting out to the point where you have that beautiful situation. It's the first time in our history that we can do this. We're actually meeting experiment. So, we can push out to milliseconds. We can look at things which are, over 100 nanometers in width. So we can actually converge with experiment. It's very exciting. So I'll show you an example here. This is ASUS's work that I mentioned already, but just to say that it's with the coarse grain modeling, we really do get out to those much, much larger scales. So we can really get out into the experimental time scales. So what SUSE does is she designs a copolyelectrolyte-based shell for Herceptin. So she's tuned the pKa of the side chains on the polymers so that it remains intact inside the body. So here, for example, at gastric pH, you get these beautiful shells of the copolyelectrolyte around your antibody. So you get these clusters of antibodies inside these shells. Now, you could never do that, well, I won't say never, but it would be incredibly difficult and time-consuming and laborsome to do this with full atomic resolution. So these are bead models. And this is programmed so that at physiological pH, thing bursts and then reforms again. So it's something that could potentially, with experiments, lead to pills for breast cancer treatment, which is exciting. Okay, and then so we get out to the larger scale. So data analytics, computational fluid dynamics. Norma showed us some of this yesterday. This is how the maths and stats team are working with Sarah Hudson's group to get more value from Raman. So Raman's a beautiful tool. It's minimally invasive. It's minimally non-destructive. And by helping to get more data and better, more precise data from the Raman, then they can help Sanofi in terms of online monitoring of the quality of their biopharmaceutics. These are Harry's designs for turbulence in bioreactors. They're beautiful. I mean, they measure mass transfer of materials inside these really, really complex geometries. And the Einstein diffusion equation, which we saw yesterday from Mikael, Einstein was winking at us, that's what links the nano and macro scales here. We can predict that, we can feed that into these types of simulations. Okay, so I hope I convinced you that we can and do model big things, and the lovely thing about that is it gives us access to large systems inside the body. So we can actually study and understand biological nanomachinery. So all that beautiful evolved complexity inside us. We can model things like ribosomes, like the proteasome, and we can even design treatments that interact with those controllably and hijack, in some cases, those machines. Paula mentioned this a little bit yesterday in her talk on bioprocessing. But we want to, I think PROTAC actually is a fantastic example of that, where people have designed small molecule drugs which go in there and actually interfere with the ubiquitin ligase pathways. So you get an opportunity to actually identify, rogue proteins and destroy them. And that promises, fantastic treatments, or much more safer, more effective anti-cancer treatments. So by pushing out in terms of, the quality of our software, how we can port it and optimize it on supercomputing centers, we get a shot at actually tackling these really, really challenging processes. So in a nutshell, I hope I kept in time, I think I probably have. So we do this because by looking at the full biological complexity, we can look at how small drugs actually interact with the body. That also gives an opportunity to develop effective peptide formulations. They can be short peptides, they're better because they're cheaper, or they can be very big peptides, they can go towards proteins, you can go towards antibodies. So we can model at all stages of that. In the future, we can push towards molecular level control of bioprocessing. So basically, we're used to looking at molecular glues, like say cyclodextrins sticking to peptides, metals sticking to peptides, proteins sticking to each other. Here at glues are those cell surface motifs, so the peptides on the surface of cells. Can we design excipients that can actually, controllably interact with those cells and maybe redirect their assembly? That's basically our challenge. Okay? Thank you.